Hello friends, this week I had posted a fairly simple MCQ on all my social media platforms. Let's see the MCQ and figure out the answer. Which of the following does not have a voltage sensing mechanism? And the options given were DHPR in the skeletal muscle, dihydropyridine receptor, DHPR, then the calcium channel in the pancreatic beta cell membrane, sodium channel at the axon hillock, and nicotinic acetylcholine receptor in the autonomic ganglia. Now the answer is fairly straightforward and one can easily guess it. Nicotinic acetylcholine receptor in the autonomic ganglia. First of all, the name itself gives you the hint. Acetylcholine receptor. So, it is going to sense the acetylcholine, the ligand, and the associated channel will open. So, it's a ligand-gated ion channel. It senses the chemical, the ligand, in this case, the acetylcholine, and it opens. It does not have a voltage sensing mechanism. What about the other three options given? So, let's see how the other three options mentioned here, they do have the voltage sensing mechanism. All right. The first one was DHPR in the skeletal muscle. Now, this is a neuromuscular junction shown here and the sarcotubular system. So, impulse, which is a voltage change, it travels through the nerve, it crosses the neuromuscular junction, and then this impulse enters the muscle, the depolarization, which is the voltage change. It travels into the muscle via T-tubule. And in the T-tubule membrane, there is this DHPR, dihydropyridine receptor. It has a voltage sensing mechanism. So it will sense this depolarization, which is the voltage change, because it has a voltage sensing mechanism. This DHPR has a voltage sensing mechanism. So it senses the arrival of depolarization and then it interacts with RYR, the ryanodine receptor, which will then release the calcium into the sarcoplasm and that will initiate the contraction. So DHPR definitely has the voltage sensing mechanism. Next option was pancreatic beta cells. In the islets of Langerhans, there are these beta cells which release the insulin. Now, for the secretion or release of insulin by exocytosis, calcium is necessary. We all know this, that the calcium is required for the process of exocytosis anywhere. So you can see in the diagram that the calcium will be required for the release of insulin by the pancreatic beta cell. Now, calcium is one ion which has to be kept very, very low intracellularly. Because calcium can be damaging if the cytosolic calcium concentration increases, it can be very damaging to the cell organelles. So, whenever calcium is required inside the cell, like in this case for the process of exocytosis, calcium will be brought into the cell from the ECF. So this is what we can see in the diagram, that the calcium comes in from outside and then it will cause exocytosis and secretion of insulin by the pancreatic beta cell. But the only question is whether this calcium channel, which is causing the calcium influx, does it have a voltage sensing mechanism? Yes, it does. So let's check out how this entire mechanism works. Now, whenever there is increase in the plasma glucose, this plasma glucose will enter the pancreatic beta cell and it will undergo glycolysis. There will be glycolysis of this glucose inside the cell, inside this pancreatic beta cell. This glycolysis will generate the ATPs. And now the important part comes up. The ATPs generated will be sensed by the potassium channel. You can see here a potassium channel, which is the ATP sensitive potassium channel. So it senses the ATPs which are generated now ATP sensitive potassium channel closes as it senses the ATPs. And since the channel is closed, the intracellular potassium, which was expected to go out via these channels, now these channels are closed. The ATP sensitive potassium channel is closed, so it cannot go out of the cell, which means what? This intracellular potassium simply comes near the membrane and starts lining up on the inner aspect of the membrane. So potassium means positive charges. It starts lining up on the inside of the membrane, and this is what is happening here. It is the change of membrane potential, change of membrane voltage. This change of membrane voltage is sensed by the voltage gated calcium channel. So this voltage gated calcium channel has that voltage sensing mechanism, and since the membrane voltage has changed now because of the accumulation of potassium, it will be sensed by the calcium channel, and the calcium channel opens. The calcium comes in from outside, and as a result the insulin will be released by exocytosis. So obviously the calcium channel has that voltage sensing mechanism. It can sense the voltage of the membrane or change of voltage of the membrane. The third option was sodium channel at the axon hillock. Now again, yes, this is a diagram which shows a neuron, and axon hillock is the portion where the axon leaves the nerve cell or arises from the nerve cell. So first neuron sends the impulse, which is the voltage change. Impulse travels here at the synapse, axodendritic synapse. So it will be transferred to the dendrite of this neuron, and then the charges will travel electrotonically. There will be travel of charges electrotonically through the nerve cell body, and those charges reach here, which is the axon hillock, this portion. Now this is a voltage change, and sensing this voltage change, these channels, sodium channels at the axon hillock will begin to open, and then depolarization will be generated, or action potential will be fired for the first time at the axon hillock. So first point actually where the action potential is generated, it is the axon hillock, and these sodium channels, they can sense the voltage change which has arrived as a result of impulse coming from the previous neuron. So even this has the voltage sensing mechanism, the sodium channel at the axon hillock. And therefore the option was, which of them does not have the voltage sensing mechanism? The answer was nicotinic acetylcholine receptor at the autonomic ganglia. Now basically ganglionic transmission is a cholinergic transmission at the autonomic ganglia. So a presynaptic neuron and a postsynaptic neuron, and there is this ganglion. So the preganglionic neuron will release acetylcholine, and on the postganglionic neuronal membrane there is this receptor, acetylcholine receptor. It has got two parts. One is the receptor portion and the other one is the channel. So when the acetylcholine binds with the receptor portion, the channel opens. This is a ligand gated channel. It does not have any voltage sensing mechanism, neither does it require so, because it requires just to sense the acetylcholine and then the channel is opened. So this is a ligand gated channel, not a voltage gated channel or voltage sensing mechanism which is provided to this particular channel. And therefore that was the answer to this MCQ which was posted on social media platforms this week.